Hi, this is Bob Lessick at the Center for Biotechnology Education at Johns Hopkins University. We will examine eukaryotic genes in this session, specifically the coding and non-coding regions of exons. Our first objective is to be able to describe what a eukaryotic gene looks like and be able to draw a model. Gene predictors distinguish between initial exons, internal exons, and terminal exons. We should be familiar with those. A non-coding exon is an exon that lies completely within either the 5' untranslated region, or 5' UTR, or entirely within the 3' untranslated region, or 3' UTR. Be able to look at a eukaryotic gene prediction and describe what it represents. Finally, be able to analyze genomic DNA in a DNA database. Although eukaryotic mRNA is usually monocistronic, it is usually processed after transcription. Large regions are removed and the remaining segments are spliced together to form the mature spliced mRNA. The regions that remain in the mature mRNA are called exons. The regions that are removed are called introns. Odd terminology. Introns are removed, but exons stay in. In the end, you have one CDS on the final spliced mRNA if it is to code for a protein. Note that the location of the start and stop codon have nothing to do with the splicing process. Here is a eukaryotic gene model. The boxes represent exons, and the lines that connect the boxes are introns. The initial transcript is continuous and goes from the start of exon 1 to the end of exon 8. This gene has 8 exons. Take out the introns and imagine the boxes now side by side. That would be the final mRNA product. The blue represents the 5' UTR and the 3' UTR. The red is the coding region. The blue to red transition represents the start codon. The subsequent red to blue transition represents the stop codon. Note that the start codon is in exon 3 and the stop codon is in exon 8. The first two exons are completely in the 5' UTR. That's very common in eukaryotic genes. Here is the human transmembrane 42 gene model. The filled-in boxes represent coding regions, and the empty boxes represent the UTRs. Note that the start codon comes very early in exon 1. The stop codon comes somewhat early in exon 3. The 5' UTR looks tiny compared to the 3' UTR for this gene. Okay, now here is the NCBI annotation. Look specifically at the mRNA and CDS locations. The mRNA includes every box, including the UTRs. The CDS is just the shaded in regions. The CDS is a subset of the mRNA. The mRNA represents the mature spliced product. You see that in the locations. The mRNA starts at 1 and the CDS starts at 10. The mRNA ends at 3749, but the CDS ends much earlier at 3265. Everything else is identical. Here's a discussion question. At what numerical locations are the introns located? Now let's examine the GenScan prediction for this genomic DNA. Note the actual verified locations from the NCBI record. And here is the prediction. First of all, GenScan will only look for the CDS, not the UTRs. However, the first exon is still wrong. It's INIT for initial exon. The second one is correct. That's INTR for internal exon. But the third exon is wrong. In fact, it predicted 4 exons. It did get the start of the third exon correct. It is at position 3125. Remember 2 things. First, gene predictors are not 100% accurate. Second, note that it is looking for the start codon in the initial exon. It got it wrong. It's looking for the stop codon in a terminal exon, and that terminal exon does not even exist. At least to our knowledge, it doesn't exist. Here's a different gene, human glycoprotein 9. We see 3 exons. However, the entire coding region is in the third exon. In fact, gene prediction, which only looks for CDS regions, would see this as a single exon gene with no splicing. Basically, it is an ORF. Here's the annotation from NCBI. It correctly shows 3 exons in the mRNA and only 1 in the CDS. Note that the first 2 exons of the mRNA are not at all part of the coding region and do not appear on the CDS line. Let's look at the GenScan prediction for glycoprotein 9. Here are the actual locations. GenScan should only look for the coding region. And since that coding region is entirely on exon 3, GenScan sees it as a single exon gene, as if there were no splicing of mRNA. It looks like GenScan correctly predicted the stop codon, but picked the wrong start codon. That happens. Here is an output from F genes H of a different gene. TSS stands for transcription start site, so the mRNA begins at position 134. There are 5 exons predicted. CDSF is for first, the CDSI regions are for internal exons, and CDSL is for last exon. The start codon is at position 1295, but we don't know if that's exon 1 of the mRNA or exon 2 or exon 3. We don't know how the 5' UTR is spliced, because gene prediction does not attempt to predict anything other than the coding regions of exons. Okay, we know that some mRNAs are spliced differently in a process called alternative splicing. You probably learned that it's a way to get more than one protein out of a single gene. You may have learned that one cell type might splice it one way to get protein A, and another cell type might splice it another way to get a different protein product, protein B. Okay, looking at the glycoprotein 9 gene, what if exon 2 were skipped? One transcript would have exons 1, 2, and 3, while an alternative transcript would only have exons 1 and 3. Well, both would make the same protein, since the coding region is in exon 3, and both transcripts contain exon 3. Why make 2 different transcripts only to produce the same protein? Good question. Stay tuned. Here's an example. Human myoglobin has 3 known transcripts. All 3 mRNAs have different lengths. The protein is the same for all 3. Since the variation is entirely in the UTR, the proteins are identical. RNA-seq is a modern technique to sequence cDNAs by modern sequencing methods. New transcripts are being discovered frequently, and some do not change the protein product. Again, why go through the trouble to splice mRNAs differently only to produce the same protein in the end? The answer, in a lot of cases, is unknown. We still have a lot to learn. It's possible that it allows for variations in translation efficiency, or that different transcripts have different turnover rates. But some of the time, we don't yet know the answer. One thing that is always the case is that the CDS is part of the mRNA. The CDS can always be said to be a subset of the mRNA. The CDS may have fewer exons, but may also have the same number of exons. Gene prediction programs only look for the CDS, and they are not always right. Finally, remember that many alternative transcripts do not change the protein, and there probably is some biological significance. Thank you and goodbye.